eukaryotic cell. So, here the second type of cells that are present in the living organisms are some living organisms which contains organized nucleus containing cells, the cells which contains organized nucleus. So, organized nucleus means, as we had already discussed, nucleus which is a complete nucleus contains nuclear membrane, nucleoplasm, chromatin material and nucleolus. So, this type of cells which contains organized nucleus are called as a eukaryotic cells, and such organisms are called as eukaryotic organisms or eukaryotes. So, in the classification part, Protista, fungi, Animalia, Plantae, all these group of organisms are the part of eukaryotic organisms, whereas Monera is the one group which is prokaryotic in their nature. So, that is the difference we find here. And when we observe the presence and absence of cell wall, so here the basic thing related to where we are able to differentiate the plant cells and animal cells. So, here we know that fungi, almost all Plantae group of organisms, and some protistans, they have cell wall, but the one which lack the cell wall are Animalia, all groups, all organisms which are the part of Animalia, and some protistans. So, this type of variation can be seen based on presence and absence of the cell wall. And when it is the Animalia and some protistans where the cell wall is absent, the outermost delimiting structure of that particular cell is a plasma membrane or the cell membrane. And of course, the fungi, Plantae and some protistans which have the cell wall, they also contains the plasma membrane just inner side to the cell wall. So, that type of deviation can be seen. And what is a cell? Cell is an organized mass of protoplasm which is surrounded by plasma membrane. That organized mass of protoplasm which is surrounded by plasma membrane is called as a cell. And it may or may not contain the cell wall. Based on that, we have already said that there is a differentiation of the group of organisms. Then where does this plant cells and animal cells are differing despite they are eukaryotic in their nature? So, here there is one more cell organelle or cell structure called as centrioles. Centrioles are present in animal cells, whereas these are absent in the plant cells. And plastids, particularly chloroplasts, are present in plant cells. So, likewise, despite of eukaryotic nature also, we can find differences between the some group of eukaryotic organisms based on some particular characters like presence and absence of cell wall, presence and absence of plastids, presence and absence of centrioles, and also the presence and absence of vacuoles. In general, in the plant cells, we can see the presence of large vacuole, but these vacuoles are generally absent in the animal cell. If they are present also, they are very small, and in some of them they are called as a contractile vacuoles which are concerned with osmoregulation function. So, in a typical plant cell, so the cell is differentiated into two parts. So, one is called as cell wall and the other part is called as protoplast. So, as we know that cell wall is the protective structure which is present outside the outermost delimiting part, and inside which we can see the protoplast. So, protoplast is the one which contains protoplasm. The protoplast contains protoplasm and plasma membrane. So, inside the protoplasm we can see the presence of the fluidous structure. That fluidous structures contains cytoplasm, and apart from the cytoplasm, so the non-protoplasmic structures are also there. That is a vacuole, and along with that one we can also see the presence of nucleus in the protoplasm. Then this cytoplasm is the one which contains a cytoplasmic matrix and cell organelles. This cytoplasmic matrix is the fluidous structure, so which is polyphasic colloidal substance. This cytoplasmic matrix is the one which contains water along with the organic and inorganic materials like amino acids, proteins, carbohydrates and different types of granular structures. Along with that one they also contains inorganic substance which are micronutrients and the macronutrients. So this polyphasic colloidal substance, that is cytoplasmic matrix, so this is otherwise called as hyaloplasm. Cytoplasmic matrix is otherwise called as hyaloplasm, and this hyaloplasm contains polyphasic colloidal substance which exists both in sol and gel forms, which exist in two forms, sol and gel forms. Generally the gel form is present in the peripheral part of the cell. Peripheral part means which is near to the plasma membrane, and generally we can consider this as a ectoplast. Ecto means which is present towards the outer side, we call it as ectoplast. And the other cytoplasmic contents or cytoplasmic matrix which is present in the middle of the cell, that part is called as endoplast. Likewise this type of variation can be observed in the nature of the cytoplasmic matrix inside a cell. And apart from this organization, so what are the other what are the other structures which are present in that one is, we had discussed about the vacuole. Generally if you take a normal eukaryotic cell, in a young or meristematic cell, the number of vacuoles are many and they are small, distributed throughout the cytoplasm. But when they become mature, when the cell become matured, all the small vacuoles will coalesce or unite to form into a large vacuole which will occupy the center part of the cell and pushing all the cytoplasm along with the nucleus towards the periphery of the cell. This type of organization is called as a primordial utricle, that can be seen in the matured cells of the plant body. Then what are the type of cell organs that we can observe in a plant cell? So before that, let's identify the different types of cell organs are there. So they have been categorized into various types based on the number of layers which are surrounding the cell organelles. So let us see, there are some double membrane cell organelles, there are some single membrane cell organelles, and there are some cell organelles or cellular structures where they completely lack the membrane around them. In a eukaryotic cell, there are some double membrane cell organelles. So those are mitochondria and plastids. Particularly in the plastids, when you consider the chloroplasts which are concerned with the photosynthesis, that is considered to be the double membrane structures present inside a eukaryotic cell. And apart from this one, the nucleus is also the cell organelle of the protoplasm which contains double membrane structure, which is considered to be the primary cell organelle or dynamic center of the cell. Then there are some single membrane cell organelles inside a cell like sperosomes, dictyosomes, peroxisomes, Golgi complex or Golgi apparatus, endoplasmic reticulum. So these are etc. some of the cell organelles which are covered with a single unit membrane, and there are some membraneless cell organelles inside a cell like ribosomes, microtubules, So actually we should not consider them as cell organelles. The cellular contents we can consider microtubules, microfilaments. In some cases the centrosome and centrioles which are present in the animal cell. So these are the structures which does not have the envelope around them. These are structures which does not have envelope around them, and there is one more structure which is present inside the nucleus, that is called as nucleolus. The nucleolus also not having any membrane around it. So this type of organizations or cellular structures are cell organs which are present in the cell. Then the cytoplasm will not remain static every time, so it will be in a dynamic state, that is always moving from one end to the other end, or some type of movements can be seen. So generally we call this type of movement as cyclosis, or here we can call it as a cytoplasmic streaming movement. This shows the dynamic nature of the cytoplasm. And here because of this cytoplasmic streaming movement, so here the cytoplasm in the form of fibers will be moving from one cell to the other cell through the plasmodesmatous. When they are moving in such a manner, so they are also helping the transportation of food particles from one cell to the another cell. This type of cytoplasmic streaming movement is always been observed in a plant cells. If you observe the diagram of a typical plant cell, where we can see the presence of various structures along with the cell organelles, cytoplasmic content, and also the other structures which are concerned with the metabolic activities of the cell. Generally there is no definite shape for the cells. So here it may be polygonal or different types of shapes can be seen, and in this one cell wall, this is a cell wall structure. In the cell wall structure we can see the presence of three parts. One is called as middle lamellum. The one which I am drawing now, it is a middle lamellum part, and on either side of this middle lamellum is the primary cell wall, or if it is a matured cell, the cell also contains the secondary cell wall, but if it is a young cell, there is no secondary cell wall in it, only primary cell wall will be present. And in the cell wall we can also see some gaps here. So these are the gaps which are present here, which are called as a plasmodesmatic connections or the pit connections between one cell to the another cell. So this is one cell, and this cell is surrounded by many cells, and those cells are binded with the help of a chemical membrane structure called as middle lamellum. And inside the middle lamellum, this part from here to here, this part is called as primary cell wall. And if it is a matured cell, so inside the primary cell wall, one more wall layer will be deposited, that is called as a secondary cell wall. And if it is a young cell, there is no secondary cell wall, but inside this primary cell wall only we can see a living membrane. This living membrane is called as a plasma membrane. Whereas cell wall is outermost delimiting structure, which is a dead membrane. This plasma membrane is just oppressed on inner side, or it is binded to the inner side of the cell wall. The cell wall is discontinuous, but the plasma membrane is continuous. So this part is called as a plasma membrane or plasma lemma. Inside the plasma membrane, we can see the different types of cell organelles along with the vacuole also. So here if it is a young cell, so the long very big nucleus will be present in the center like this. This is a nucleus present in the center in a young cell. So this nucleus contains a spherical shaped darkly stained part inside that is called as a nucleolus. The number of nucleolus may be 1 to many, and this is a nuclear membrane. This part, all this layer is a nuclear membrane. And nuclear membrane is also discontinuous, which contains a pore, that those are called as nuclear pores, and inside which we can see the fluid matrix. This fluid matrix is called as a nucleoplasm. Apart from this one, we can see thread-like irregular mass, irregular structure. So this thread-like irregular structure is called as a chromatin material. That is nothing but a chromosome. Later it will be it is condensed and formed into chromosome structure. And here, if you could see in this diagram, outer layer of this nuclear membrane is continuous to form into some membranous folding like structures. This membranous folding like structure is called as endoplasmic reticulum. And here and there we can see the presence of some stalk like group of mass of structures. This type of structures are called as dictyosomes or Golgi complex. Here and there we can see many such structures. Then apart from that one, we can also see some other cell organelles like chloroplast, which is a double membrane structure. Along with the chloroplast, mitochondria can also be seen here and there inside the cell. This is also a double membrane structure. This forms a mitochondria. And somewhere here the endoplasmic reticulum is binded with some granular structures. So these granules may be sometimes freely present in the cytoplasm, or sometimes they may be studded to the endoplasmic reticulum. So these spherical shaped structures are called as ribosomes. These are membraneless protein synthesizing structures. And here and there we can see long and elongated structures like this. These are called as microfilaments or the microtubules. Apart from that one, so we can also see the presence of an empty space here. That empty space is called as a vacuole. And this vacuole is surrounded with a membrane called as a tonoplast. So these are the structures generally we find in a cell. Apart from that one, in a eukaryotic cell we can see the various types of starch grains or glycogen granules, which are nothing but a type of stored food materials present inside the cell. So apart from that one, peroxisomes, spherosomes, dictyosomes, this type of micro body structures are also being observed inside a typical eukaryotic plant cell. But here centrioles are completely absent. In place of that one, the microtubules and the microfilaments are present. In a plant, typical plant cell, the first part is a cell wall. Cell wall is the outermost delimiting structure which is non-living. This is the one which is secreted by the living matrix present inside the cell. So living matrix means the protoplasmic contents which are protoplasm which is present inside the cell is secreting the cell wall. And this is hard, rigid and tough part. So these are the characters related to the primary characters related to the cell wall, and this cell wall is one which is having the chemical composition of different types of materials, which is made up of lipo, which is made up of cellulose, hemicellulose and suberin, lignin like substances. So, in the chemical composition, the first part of the cell wall is a matrix which contains water about 60%, hemicellulose of 5 to 15%, pectin material which is of 2 to 8%, protein which is of 1 to 2%. Next one, lipids of 0.5 to 3%. So, this is a chemical composition of the matrix of the cell wall. Then apart from this one, this also contains the other ingredients. So, what are the other ingredients which are present in it? Suberin, lignin, cutin, and some others will have the silicon like different types of substances are also present. So, after matrix, apart from these other ingredients, it also contains the cellulosic microfibrils. So, in this the major composition is the cellulose only. So, which is of nearly 10 to 15% of its composition. So, this is a cell wall composition where matrix, cellulosic microfibrils and other ingredients are present. In the matrix, these are the various types of chemical substances and their proportion of their percentage, and in the microfibrils the major composition is of cellulose only. Then other ingredients which are acquiring the present in a very trace amounts in a cell wall. So, let us discuss the other characters related to the cell wall. How does the cell wall is helping in the cell from various other activities. So, first one, as I said, that is a rough and rigid structure. So, this is providing shape to the definite shape to the cell, and it is concerned with protection from various types of organic and inorganic substances, and also protection from the poisonous or toxic substances, and also it is concerned with protection from osmotic lysis. So, like when the endosmosis occurs, when the water goes on entering into the cell, what happens? The volume of the cell will decreases, but here the cell wall will delimit the endosmotic process, so that it is protecting the cell from the osmolytic lysis. Then it will provide rigidity and toughness. This rigidity toughness is due to presence of lignin material in the secondary cell wall, and sometimes they may be impermeable to water also. Actually, when does they will be impermeable means? So, they will be impermeable to water only when the chemical called as suberin is present in the cell wall. But actually, cell wall is a permeable structure, as it is not having the ability to select only some substance to come inside and some substance to go outside. So, it is actually a permeable membrane, but whenever the suberin is present, it is impermeable to the water. So, here during the osmotic process or the passively when the water is coming into the cell. So, first the water or whatever the components which are entering into the cell, they will pass through the cell wall and come into the space that is present between the cell wall and the plasma membrane. And apart from this one, the cell wall will be the part of apoplast in a cell. Then what is apoplast here? Apoplast is an continuous system which is containing cell wall and intercellular spaces. As we said that intercellular space is an empty space, so it is a non-living structure. In the same way, cell wall is also a non-living structure. So, apoplast is a continuous structure of non-living structures, that is cell wall and the intercellular spaces. And of course, as it is not like that, as a non-living they are not concerned with any function, they are helping in the apoplastic movement of water. That means sometimes the water will move from one cell to other cell through that cell wall and the intercellular spaces. So, they are concerned with the transportation of water and also the minerals, so that those substances are carried from one cell to the another cell. Basically cell wall is differentiated into 3 layers. So those are called as middle lamellum, primary cell wall and secondary cell wall. Then starting with the first one, the middle lamellum or middle lamella we can call it as. When does this middle lamellum is formed in a cell? So this is a part which is formed during cytokinesis. So that is during the process of cell division, when a cell is dividing, first the nucleus will divide to form into two nuclei, two daughter nuclei, that is karyokinesis we call. After that the cytoplasm will also start dividing, or we can call the cell is dividing, that is called as cytokinesis. During the cytokinesis, the middle lamellum is formed, and this middle lamellum is acting as a cementing like substance. So or it is acting as a glue like substance. So it is a structure, or it is a structure which is concerned with binding of the two adjacent cells. Consider likewise a group of cells are present like this. So these group of cells are binded with the help of this glue like substance called as a middle lamellum. So that when it is acting as a glue like substance, it will not allow the movement of the cells, it will not allow the cells to get separated out. So that particular substance, cementing like substance called as middle lamellum, is helping the cells to hold in one particular position. So that is the significance of this middle lamellum, and middle lamellum is considered to be the first layer formed in a cell. And consider from this part to this part. So this is a middle lamellum part which is common for these two adjacent cells, and from here to here this is a middle lamellum part which is common for these two cells. In the same way, this is from here to here, this is a middle lamellum part which is common to these two cells. That means middle lamellum is not an individual layer of a cell, it is a common cementing like substance present between any two adjacent cells. Then what is the chemical composition of this middle lamellum? It is made up of calcium and magnesium pectates. So pectin is an organic substance which is present in the middle lamellum, whereas calcium and magnesium are the inorganic substances, and these two are considered to be the macronutrients or macro elements. Calcium and magnesium are the macro elements which are present in the cell wall of which are present in the middle lamellum chemical composition part. This is the first structure present in cell wall called as a middle lamellum, and the middle lamellum is always continuous, because in some places the primary cell wall and second cell wall are discontinuous, but at that region also this part will be a continuous structure. The second part here in the cell wall is the primary cell wall. Primary cell wall is the first layer of, or first individual layer of a cell, because I say that middle lamellum is the first layer, but it is not an individual layer for each and every cell, but primary cell wall is an individual layer for each and every cell. Then consider that the middle lamellum is there like this. Likewise, this part we consider this as a middle lamellum of a cell, and of course it is a common for all the adjacent cells. And inner side to the middle lamellum, a layer is formed, that is called as a primary cell wall. So it is always formed towards inner side to inner side of middle lamellum. And primary cell wall is present in young cells. So young cells like meristematic cells or meristems, young cells like the parenchyma cells, the mesophyll cells, where the second cell wall is not deposited, and this primary cell wall is generally little bit flexible also, or we can say elastic property is there. And when we say that cell is increasing its volume, that is because of the elastic nature of this primary cell wall. And if you see the chemical composition of a primary cell wall, chemical composition of a primary cell wall, here cellulosic microfibrils are present. So these cellulosic microfibrils are present in the pectinous matrix. Pectinous matrix, and these are the two major chemicals which are present in the primary cell wall. Apart from that one, hemicellulose is also present. So these are the three major chemicals. In that pectin is present in large quantity. So this is in the pectinous matrix, here and there we can see small empty spaces or unfilled spaces are there. Those unfilled spaces are filled with the cellulosic microfibrils. This type of layer is deposited on the inner side of the middle lamellum called as a primary cell wall. And here in the primary cell wall, here and there we can see some empty spaces or the thin areas are present. These thin areas are called as primary pit fields. So why such thin areas are formed is when the deposition of the primary cell wall material is taking place, in some areas the primary cell wall is not deposited. So that is acting as a connection between one cell to the surrounding adjacent cells. So here through this primary pit fields, the cytoplasm will be moving. The cytoplasm will be moving in the form of strands. So the cytoplasmic strands which are moving through the primary pit fields along with the primary pit fields is called as plasmodesmata. The primary pit fields along with the cytoplasmic strands which are moving from one cell to adjacent cell is called as plasmodesmata, and these plasmodesmata are acting as a connecting link between the two adjacent cells, and it is concerned with the transport of materials from one cell to the adjacent cell. So these are acting as intercellular transportation channels. And these plasmodesmata are surrounded by plasma membrane, because inside the primary cell wall the plasma membrane will be there. So here the plasma membrane is covering the plasmodesmata. So on either side also the same thing. On the adjacent cell, this is the primary cell wall of the adjacent cell, and the inner side to that one the plasma membrane is present. So that plasma membrane is a structure which is present on either side of the plasmodesmatic structures. So this type of special structures are present in the cell which are concerned with the transportation of materials from one cell to the another cell. So that is called as plasmodesmata. The proteins which are present in the primary cell wall is formed by a process called as intussusception. Intussusception means the process of formation of a layer, new structure in the form of a complete layers is called as intussusception. All the time the new layers will be formed during the primary cell wall formation. So we call this process as a intussusception process. Okay, next one is secondary cell wall. Secondary cell wall may not be present in all the cells, because it is present only in the cell when it is in a matured condition. So like only the matured cells contain the secondary cell wall. This secondary cell wall is always deposited on inner side of primary cell wall. Then this secondary cell wall is the structure which is providing rigidity and toughness to the cell. Then here these secondary cell wall is present in the cells like tracheids, vessels, collenchyma, sclerenchyma tissue. So in such type of cells we can see the presence of secondary cell wall, and secondary cell wall is present in matured living cells and also dead matured cells also. In both of them they are present. and actually when the new layers of second cell wall is going or depositing, the lumen size, the size of the protoplasmic content is gradually being decreased. So this type of nature will be there, and second cell wall is similar to the primary cell wall in its chemical composition to some extent. That is, here it is also having cellulosic microfibrils. This is a common feature that is present in both primary cell wall and the secondary cell wall, but here the unfilled structures in the cellulosic microfibrils are filled with various other types of materials, that is either it may be suberin, lignin, cutin. In some of them it may be with the silicon, like silicon dioxide like substances. So whenever the suberin is present, it is providing impermeable nature. Lignin is the one which is providing toughness to the cell, and whenever the cutin is present, so here cutin is the one which will reduce the rate of transpiration, as we know that is present when in the form of a cuticle layer in a exposed part or aerial parts of the plant body. So likewise these are the structures, characters related to this one, and here always the secondary cell wall is deposited in the form of layers, and this how many layers are present in a second cell wall means generally many layers are present, but basically there are, in majority of the plants the secondary cell wall contains 3 layers. So those are known as S1, S2 and S3 layers. When the number of layers are more, the numbers will be continued like Sx we can consider. Likewise the number of layers will goes on increases, and S1 is the first layer which is formed just inner side to the primary cell wall, and inner side to S1 layer, S2 layer will be formed, and inner side to S2 layer, the S3 layer will be formed. Likewise the layers of a secondary cell wall will be formed when the cell is getting older and older. So in that process, consider this as a cell where unfilled places are also there, which are called as pits. As like we had said that primary pit fields are the thin areas present in the primary cell wall. In the same way, the unfilled or thin areas are present in the second cell wall, those are called as pits. Actually here they are acting as connections between the two adjacent cells. Here these pits are of two types, one is called as simple pits and other is called as bordered pits. So here let us see the structure of the simple pits. So if you observe from outside, the simple pit appears to be like this is a cell wall which appears to be like a single structure like this, spherical shape structure. So here if you take the section of this one, section of the cell wall, so this is the middle lamellum present here in the center, and inner side of the middle lamellum, this forms the primary cell wall. So which is also formed nearly continuously, but sometimes it may form the pits also. So likewise continuous layer may be formed, or sometimes the pit containing structure, and on that we can see the presence of this type of secondary cell wall deposition, and throughout its length from this region to this region, the thickness of the secondary cell wall will be remain uniform. So that is the reason why if you see the cell wall pits, or if you see the structure of the pits on the cell wall, it appears to be spherical in their nature. So this is round structure will be formed. So uniform thickness is present in the secondary cell wall in the simple pits, whereas in the bordered pits the thickness of the secondary cell wall is not uniform, and on outer side we can see from outer view the cell wall which is having this type of organization on its wall. So here it appears to be like a border like structure, that is pit of one cell and a pit of the adjacent cell, both of them appearing to be on the cell. So here always the bordered pits are present in pairs, present in pit pairs, and they forms a function unit. One single function unit will be formed by two simple pits, and here if you see the internal organization, this is the middle lamellum which is continuous, and on the middle lamellum the primary cell wall is formed. So this is also sometimes continuous or discontinuous. If it is discontinuous also, the thickness will be uniform in the primary cell wall. But when it comes to the secondary cell wall, so here the thickness is less. The thickness is showing the thickness of this second cell wall is differing. So here from the, if you observe in this region, this region, this region and this region, the thickness will be different. So this region thickness is less. So if you observe, the thickness will be changing from one side to the other end. So here also the same thing. And the second cell wall is deposited in the form of an arch-like structure on both the sides like this. Arch-like structures. And this is a structure which is actually the middle lamellum, and we call it as pit membrane. Middle lamellum, this part is called as pit membrane, and sometimes the primary cell wall is also there, but sometimes it may not be present. And here this empty space which is present inside this arch-like structure is called as a pit chamber. And this arch-like structure which is bending, this part is this part, this is called as a pit border. Then the empty space which is left out here, this is called as a pit aperture, this region from here to here, on this side from here to here. So this is called as a pit aperture. So these are the structures that can be observed in the bordered pit. And here if you take the section, so the thickness is different from this region to this region. So this shows that the border like structure is present. This type of pits are called as a bordered pits, present in the pits of some of the plants in the secondary cell wall.